Good afternoon, and thank you, Michael, for the opportunity to moderate this afternoon's session. Those of you that were with us this morning, thank you. Those of you that were with us this morning, we had a dynamic group discussion this morning, and hopefully it will continue this afternoon. I have the distinct and unique pleasure of introducing our first guest speaker this afternoon, and that's Dr. Carl Dieffenbach. And I think I have the unique pleasure also of being able to call Carl both a friend and a colleague. I've known Carl for decades, and I have had the distinct honor of being able to call on him to ask him questions when I've not known something, been able to rely on him for very direct advice when I've been lost and struggling with understanding of what to do from a unique perspective as a community member, and I've always relied on him to give me an honest, earnest, and scientific response. So I'd like to introduce Dr. Carl Dieffenbach, who is the director of the Division of AIDS at the National Institutes of Allergies and Infectious Disease, NIAID. Carl?
Thank you. It's a pleasure to be here. And just a word about Tony. She was an absolutely important person as we got started in our Washington, D.C. initiative. Gave us sound advice and has been a critical sounding board for us every step of the way. The first thing she said is, don't make people go to the NIH, to the building, because we're surrounded by a fence and it just sends the wrong message. Move your clinics into town. And that was the best advice we could have had. And so I thank her for her stewardship and leadership in the community in Washington for the past 15, 20 years. So today, what I thought I would do is give a little bit of a primer on cure and talk about it from the standpoint of what does cure mean? What does it mean to cure a virus infection, and use, in addition to HIV, 2 other examples. But then talk about something else that I think is really important for us to all to understand, and that is, how do we measure? What are the critical measurements we need to make in HIV? What have been the game changers for assays that have come along that have made a difference in our success in fighting HIV, and why I'm particularly hopeful about where we are today before I get into much of what you've heard already today about where the field is. So Merriam-Webster defines cure as the complete or permanent solution or remedy to bring about recovery from or to permanently restore health or soundness. I think that is something that we all would like to be able to achieve for HIV. So what happens with that when we look at it from the perspective of the virus infections, flu, hepatitis C, and with HIV? So on your right, as you're looking at the screen, is the graph of the flu epidemics that occur every year. And as ceaseless as the tide, flu comes annually. And while we have vaccines, they're usually anywhere from 20 to 50% effective. And the lower panel shows the significant pandemics where millions of people have died. Flu is not thought to be a big deal. However, if you get it, you know it's a big deal. It is an incredibly infectious agent. If it's uncomplicated flu after exposure, you have a short incubation, intense fever, and weakness and fatigue up to weeks at a time. And then the other concern after you've recovered is, as flu evolves, you get to get sick again because flu changes. The current epidemic that we're undergoing with flu is not on this map, but actually is tracking the The blue line here, almost exactly. So in terms of a year, it's the equivalent of the case rate is equivalent to what we saw in 16 and 17. This is where the idea, you've seen a lot in the news about universal flu vaccine, and there's a lot of work going on that right now. And if we were able to get a universal flu vaccine, we would drop the case rate fairly significantly. So here's an example of an infection that is readily controlled by humans pretty well. However, it does recur because of the variation. So the one thing that's critical to all three of these viruses is variation is a theme. The natural variation in the viruses is a theme. So another virus that is also a significant public health challenge is hepatitis C, or HCV. It's important to note that between 15 and 25% of people who become HCV positive actually clear the infection on their own within the first 6 months, indicating that there is a role for immunity here that matters. Early in infection, just about everybody remains asymptomatic, and progression to liver disease takes a significant amount of time. And we have been able to generate a body of new medications called direct-acting antiretrovirals, which creates a cure in greater than 90% of people who complete treatment that can last anywhere from as little as 8 to as much as 12 weeks. So this is a very significant advance. Fundamentally, however, if you are cured via drug treatment, reinfection remains possible. And so there is no natural immunity that builds up with infection against hepatitis C. So in many ways, like flu, even though when you're cured, reinfection is possible. And it may have to do also with the level of the lack of ability to generate immunity. Moving on to the virus that makes us, why we're all here today, what makes HIV unique from these other two? Now, a central tenet in human immunology, mouse immunology, mammalian immunology, is the purpose of the immune system is to activate and respond to an infection. It could be seen as a challenge to the immune system. The body responds to the challenge and creates a response that pretty much most of the time clears that challenge, creates memory and rest, and goes back to sleep. So think of it as a fire truck that is tailored to that specific infection. It goes and puts the fire out and parks itself somewhere in the body so that at some point in the future, if the body is re-exposed to that same insult or agent, that fire truck can come out, activate, and respond. However, what HIV does is it integrates, it becomes part of the cell that is essential for that response. And as such, as soon as that cell reactivates in response to further stimulation, it further stimulates the growth of HIV. So HIV has found a way to exploit the very core tenet of immunology for its own nefarious purposes. And therefore, it remains the significant challenge that it is today. And because it becomes part of the cellular DNA is why we actually need to talk about a cure. Because even with antiretroviral therapy, if you think about what Merriam-Webster says, a complete or permanent solution or a remedy, or to permanently restore health. Antiretroviral therapy restores health, but not on a permanent basis, because as we are all well aware, it requires, for now, at least one pill once a day. So why do I care about measurement? And why should you care about measurement? And why should you be aware of what assays or technologies have come along that have made a difference in HIV? I think it's important for us to understand where we have been and therefore use those as guideposts for where we need to go in the future as we progress toward a better HIV vaccine, better prevention, and what we need to do for an HIV cure. And not surprisingly, the first assay that came along was the blood test, which allowed us to differentiate who had become infected with HIV and who was not. It allowed governments around the world to protect the blood supply, to diagnose those particularly early in disease, because as we are well aware, frank AIDS was obvious, but who actually was going to progress to AIDS was not known. But it wasn't until we had first the blood test, and it wasn't until we had a blood test that we were able to then figure out that the CD4 test would be a good marker for disease progression. It also demonstrated the extent of an asymptomatic period and was essential for early epidemiologic and natural history studies. So very early in the epidemic, this allowed us to help begin to define what HIV was and what HIV wasn't. The next test that came along had this relatively complex, fairly dense, and not very easy to understand title from a person who many of you may have heard of, Jeff Lifson, who is a leading scientist at the National Cancer Institute. What this was the very first description of using polymerase chain reaction in a quantitative way to measure the amount of virus in plasma. So this was the first example of quantitative plasma virus measurements. We all know and speak routinely now about viral loads, but this was the first demonstration that viral load actually mattered. And the title of the paper is quite important: High Levels of HIV in Plasma During All Stages of Infection. And that was the message, there's no such thing as a silent infection here. It is persistent, it's ongoing, the virus grows every day, every hour, all the time in the human body, and it needs to be dealt with in that kind of a way. How did this advance the field? Very quickly, from 1993 on, the first groups led by David Ho and Marty Markowitz, and then George Shaw and his team, were able to demonstrate that the drugs we were working on at that time actually had an impact on plasma viral load. And David and George's group demonstrated the test of concept that when you provide a medication to an individual and their viral load starts to drop, you can literally measure it in days to hours, and it didn't need weeks or months or years. And that gave us the ability to think about viral load ultimately as a surrogate, and that's why, in many ways, this initial finding got us to where we are today with U equals U. Because without the ability to say somebody is undetectable, therefore, or they cannot transmit, we would not be where we are today. So this is why these kinds of assays truly matter. Now, where are we with assays on the cure? So this paper was just published from Catherine Bruner in Bob Silicano's lab, again with an important title of Quantitative Approach to Measuring the Viral Reservoir of Latent HIV Proviruses. And I hope in 5 years I can come back to an event like this and I can say to you, this was the paper that has made a difference in cure. Because I think this assay has some specific characteristics of it which we will be able to see if it is capable of what I hope it's capable of being able to be the equivalent of measuring viral load, but this will be the assay for the cure. And it is a very simple assay. First and foremost, it takes into account that the vast majority of the DNA that is present in our bodies when we're on therapy that we're carrying in our CD4 cells, the proviral DNA is defective. And only about 1 in 1,000 of those is in fact completely intact. And what Bob's lab was able to define was a region here called the size site, which is also known as the packaging site, and a region embedded in the middle of the envelope are highly conserved. And in a very simple way, it's essentially 2 PCR reactions. So again, PCR is going to play a major role. But the output of the assay is brilliant. It essentially looks like a flow diagram. Think about it as remember Foursquare when we were kids? And the goal was always to get into the 4th square, so you were ahead serve. So Square one, which is also known as quadrant 3, is negative DNA, no provirus present. Quadrant 1 is one of the two regions is present. Quadrant 4 is the other segment is present. Quadrant 2 is both segments are present, indicating that any dot in this quadrant indicates an intact provirus. Very simple, very easy. And as such, then, is a very rapid readout and a significant improvement over the assays we currently have. Importantly, this has worked well in terms of measurements, and the group, as part of this paper, was able to show that by measuring the intact virus, and the presence of one of the other two markers had different patterns of behavior within individuals over time, indicating that there was, as you could see, significant fluctuation in the other two and a degree of stability in the intact provirus. This is important now if we can get some tools that have an impact on the intact provirus. With this kind of a very simple assay that can be run in a day, we'll be able to see a drop, not over a course of years, but days to weeks. So that's why I'm excited about this assay and wanted to share my enthusiasm and hope that I'm right about this, and you can come back and tell me years in the future, if it's not, that I was full of shit. So where are we today in our path toward a sustained art-free remission for HIV? And in many ways, there's two paths we can take. We can talk about eradication of the competent HIV reservoir or a classic cure, or we can talk about sustained virologic remission, which would allow us to create a level of immune-based control that would prevent viral rebound without in the absence of antiretroviral therapy. And as a famous baseball player who played for the Yankees once said, Yogi Berra, if you come to a fork in the road, take it. And so we're going to go through both. So let's talk first about what eradication would look like. And what we have in the audience with us today, celebrating Timothy Ray Brown and his 12 years, is an homage to him, in that there is still so much we can learn from Timothy and his case. That is that Timothy was not only treated for one disease, but he was treated for two. And in some ways, as you can see on the graph here, in Timothy's story we've all heard, is he had to go through 2 rounds of chemotherapy and 2 rounds of immune reconstitution. HIV was taken care of by the first, but his leukemia required a second. So for patients like Timothy that have 2 diseases, cancer and HIV, cancer is probably the worst of the 2, and his leukemia, as it roared back, required literally a sledgehammer-like approach to help him get through his lethal cancer. Apparently, at this meeting, there's an example of another patient that has had a stem cell-based full remission, and I have not seen the data, and we all look forward to seeing the results at CROI for that. So again, congratulations, Timothy. It's great to have you here for your 12th anniversary. There are a range of strategies that can be used and stem cell transplantation is what we've just discussed. But the significant work has been going on recently using CRISPR-Cas9 and other strategies for gene editing. There are modified antibodies and then latency reversing agents. So to steal a page from the DefeatHIV, one of their cartoons from one of their papers, this is the places where there are multiple targets for anti-HIV gene therapy, and I've highlighted a couple on this slide. As you can see let's see if I can get the arrow to work. With this arrow, it's not showing up on the screen. The arrow on the upper right is the use of vectored antibodies. Can you take antibodies or other biologics, put them in some sort of a gene therapy vector, put them in the body, and so that the vector will then lead to the production of this protein, this molecule that will be in the body that will serve to protect the cells from the spread of HIV. Currently, we're using broad neutralizing monoclonal antibodies for prevention through studies like the AMP trial that the HIV vaccine trials network and the HIV prevention trials network are performing, but it's being used also by others in more therapeutic and antibodies are also being used in therapy as well. But the idea would be not just to give the antibodies, but actually to give the antibodies in a way that the body took care of producing as a drug inside. The other is there's at least one talk at the meeting as well this week on engineering cells so they are genetically resistant to HIV by eliminating the co-receptor, as labeled here by the delta CCR5 T cells. There are several groups that have been using genetic technology to go in and edit cells so that they lack the co-receptor so they cannot be infected with HIV. And last but not least is the idea of could you use these technologies to go in and clip out the provirus directly in a way that eliminated the latent virus reservoir from the cells in the body. There's a lot going on in these areas. And at this point, it is still, in many ways, ex vivo. We have not quite figured out, and we probably won't for another 10 or 12 years, to how to give it these kinds of technologies as a shot to an individual. But ultimately, that's the kind of thinking we need to get to, is do this in incremental steps, first so it's safe, and build the evidence base and move carefully one step at a time to where it becomes a single shot and we can then have something that can truly make a difference in people's lives. So moving on to the other pathway, which is sustained virologic remission in the absence of eradication. The reason this is somewhat more attractive at this point in time is this is the kind of a solution that could be low risk to the patient. It could be scalable and could potentially lead to induction of a durable immune-mediated control of the virus. So the kinds of things that we could do could be the kinds of things that would require an intermittent type of delivery of, say, monoclonal antibodies, say once yearly, if we could get to a series of long-acting, or we could get to a point where we truly induced changes in immunity that required no further intervention. I think the challenge with these things are the need to provide to people who are in an ART-free remission time, where they have low or no detectable viral load, is the tools to self-test for when they would rebound. And I think that we really need that technology to catch up with us to where in the privacy of your own home or apartment, that you could do a dipstick, you could take a drop of blood and do a single test that told you whether you had rebounded or not. Because I think that's the kind of technology that's going to need to keep up. Also, there's a lot of community interaction that needs to go on in this space, because we need informed individuals that understand that they are participating in an endeavor of extreme importance. And it's then a pact between the doctors and the study participants that we all have a role to assure the safety of the participant, but we also need to give them the tools as well as the knowledge base so that they understand what they are participating in. So this is a slide from a very old paper that is now over 20 years old from Rick Davies' group at NIH, where they took a number of people who had been on antiretroviral therapy for several years and did one of the first structured treatment interruptions. And you can see the vast majority of people in the study rebounded very quickly. But one patient, patient 18 in the study, appeared capable of maintaining significant control of virus replication following discontinuation of drug therapy. How can we make this, instead of 1 in 20, make it 20 out of 20 of people that can have this kind of pattern and control? We have to understand what makes this individual unique, and what are is it a genetic activity like what we've seen in certain individuals, certain HLA types, or is there some other component to this individual's immune system that makes them special? But we have to then figure out how to exploit that and bring it to a scale that it would benefit the other 20 people that participated in this kind of a study. So as we think about art-free remission, let's move on and talk about therapeutic vaccination and then talk about passive transfer of broad neutralizing monoclonal antibodies. So a therapeutic vaccine remains in some ways a holy grail. There has been lots of attempts at therapeutic vaccination. None have had anything more than a statistically significant drop in viral load, nothing that has been truly useful to an individual, but it remains an area of emphasis. So I took the liberty of modifying this slide and added the blue lines there at the bottom, because that's my fantasy, that as people are vaccinated and then stay off therapy, that there continues to be profound immune response that continues to drop the level of DNA present in the blood and in the tissue of an individual. And then with rebound, you would have essentially no rebound. That is the dream. So there's been at least one study that had some level of effect like this using therapeutic vaccination, using an Ad26 MVA therapeutic vaccination. This is very similar to the vaccine that is currently being used by the HVTN in the 705 trial for HIV prevention. That trial, the 705, uses Ad26 plus protein. The company that was working on this, Janssen, had looked at combinations of Ad26 and MVA for prevention, and they're very interested in using a combination of 26 and MVA in therapeutics. But what this was able to show is that the combination of this with a specific activator of immunity called TLR7, which is actually a strong inducer of interferon, it had an effect in rhesus macaques. So here's an example of a different molecule that was delivered via gene therapy. And I believe you saw some of this data this morning. The folks from Scripps, Florida, were at least on the agenda, and talked about ECD4IG as a molecule that could be delivered via a virus vector called adeno-associated virus. And this is one experiment that they have talked about publicly in the past, where animals were given AAV or control, and you can see in the animals that received the AAV and produced ECD4 in the blood, were able to ultimately control their viremia in the absence of therapy in a pretty profound way. So this is a very powerful molecule. There are human forms that are being made, and whether or not this is a part of an ultimate treatment or not remains to be seen, but it is promising at this time. So in conclusion, I wanted to leave some time for questions. I think we are approaching the end of the beginning. We're not in a point now where there's any clear path toward a cure, regardless of which branch, fork in the highway you've taken, but there's some promising leads out there. Do we have this new assay? Will it be dynamic enough? Will it be the assay that we have been seeking. As I said, there are hints of success that are appearing. We will continue to build on our successes, and above all, we will also learn from our failures. But together, researchers and community working together will ultimately get us to where we can succeed. So thank you. So it looks like we have about 10 minutes for questions.
Yeah. Anybody have questions for?
There's one right there.
We have one on the front row.
You mentioned the possibility of a cure being reported at this workshop. Can you tell us when that's going to be? Which session?
It's a late breaker. I don't know. What day is it? Tuesday. It's called stem cell therapy and something or other. And it's just, voices.
Thank you so much for the presentation. I have two quick questions. Actually, one is not a question. Let me begin with it. I want to thank you as NIH and your team. For the last 3 years, you have really tried to involve Africa into the ongoing cure research. And it's really good for Africa that now you're beginning to involve us into the ongoing cure research. Several research is now beginning to take place in Africa, and we pray that you continue involving us. The question is, you as a researcher, what are the promising intermittent non-art interventions that you think will help us get to the functional cure?
So I think for right now, the things that are most promising are combinations of monoclonal antibodies and therapeutic vaccines. I think that the kinds of treatments for eradication are still very, very early days. And I think we'll need to continue to look at some of the gene therapy activities more in cell culture first before we start headed in the direction of putting things directly in people. So those are the kinds of things that, as you are well aware, when we started the AMP trial, we went not just to the United States, but also directly to Africa at the same time. So these are the kinds of things that, with the antibodies, that could immediately begin with global trials, not just limited to the United States. And we've got 2 over here, too.
Okay. Hi, Carl. So, I'm wondering that you guys have been loving these monoclonal antibodies for a while, and first we thought it would be 1, and then 2, and then 3, and maybe once a year, and now once every quarter. I mean, they're going to be so expensive. I mean, I can't help to think about the nitty-gritty stuff that comes after. And so what's going to make that any different than the new long-acting other drugs that we have in the pipeline? I mean, the price is really what makes me feel like there might be a chilling effect.
So whenever I talk about these, you are in my ear. And one of the things we are continually talking about is understanding what the cost will be and making sure it is competitive with PEPFAR-type pricing. I think it has to be.
Hello. My name is Mario. I'm from Cuba. I'm wondering, because my background is from Spain, from Barcelona. I have the CCR5. I live with HIV for over 38 years, and I'm doing well. I'm wondering if it's possible to do that test, or how the doctor knows if I have this CCR5.
Are you asking how many people have those kinds of mutations?
No, I'm asking if I can do the test for figure it out if I have the...
There was a presentation this morning around CCR5. He's basically asking, how does he get the test to see if he can get in that trial?
Since I wasn't there, I don't know, but there are. Is it an NIH trial?
Yeah.
Okay.
But you work at the NIH,
I
don't
do
you?
work at the NIH. So there are, I'm going to let Katie take this one because I don't know the answer. There are specific tests that you can have to know whether you have the mutation and or would be eligible to participate in such studies. I would say talk to your doctor.
Right, and I think that we can figure out how you can, who it is that, because there was somebody who spoke specifically about the CCR5 trial, and we can find out who that person is and get you in touch with them.
Okay.
So we can
The
talk to
group at
Michael.
Penn was doing a lot of that work.
Right, and we can talk to Michael and figure out how to get you in contact with that person. I think that's the easiest way to do that, and
Yeah.
get you off the hook.
Well, thank you. Thank you.
Hi, I'm Corinne, UNC. Where do you see behavioral and social science fitting in the HIV cure research agenda?
Where do you see what?
Behavioral and social sciences.
So I think that behavioral and social science is driving the discussion between community so that we have understanding of people's perceptions. And I think that is a critical component going forward. Also, to continue to talk about the ethics of what we are doing, particularly as we start moving towards scale. That's why I mentioned the business of the need for certain types of measurements that are available to people. But yeah, clearly then, behavioral and social science would need to be able to help with the teaching, but teaching's two ways. Teach the scientists as well as teach community.
And is there funding dedicated to make that happen?
There remains grants available for behavioral and social science research through NIMH, through Child Health, and NIDA for cure-type activities. NIDA, yes, drug, because of the ability to do cure in the presence of drug use.
So my question is kind of cure-related. I really am very excited about all this research. I think that something will happen at some point, some sort of functional cure or something, but I don't think it's going to apply to somebody like me with a long, long history and a deeply, deeply embedded reservoir, and I'm kind of old now. That said, I'm very excited about it. I want to see it all move forward. But part of this is also that, you know, we have these long-acting injectables coming. We have these pills that are really easy to take. We have fabulous therapy. It's not that bad living a life with HIV except for the chronic inflammation. And if there was some way that for people like me and others, you know, to sort of shut that down to prevent these comorbidities and the premature aging and all of that, because your immune system is mounting this constant response. So I would think, hopefully, in all of this research, maybe we can figure out some way or something will surface to sort of silence the body's recognition of this virus and or, you know, other methods on the chronic inflammation.
Thank you for that comment. I couldn't agree with you more. I think what is left behind with antiretroviral therapy are these defective viruses that are essentially every cell carrying, whether it's a defective provirus or an intact provirus, literally spits out stuff all the time as it activates. And so it is acting as a flare. So if you think about what I said about immune activation, is HIV lives for immune activation and actually always leaves the embers behind to maintain that. So as we get ready to eliminate the intact provirus, we also need to be able to clear the junk behind. And if you think about what you're talking about, the level of immune activation, for people who are elite suppressors, they have very high level of immune activation and have, in many ways, more comorbidities than people just on therapy. So it is a significant problem. We have tried many different tools to try to knock down immune activation without success. It's not simple. If it were simple, we'd be done with that part of it.
Well, you might have answered my question with his response, but I was going to ask you about in terms of curing the virus, is it dynamic? Is information coming out, you say, like a measured pace, or are there really kind of signs of, here we found a lot of information about how to cure a virus, and we're letting it out to you, and then it takes another like 15 years? I mean, would you say it's dynamic research, or it's really kind of long and drawn out?
Both. There's occasional, like many fields, there are incremental progress all the time. And occasionally you take a big leap. That's the kind of thing that we just need to plan for, because we will continue with the incremental and get there. But somebody, that's the beauty of research, is there will be somebody.
I should have said I'm a doctor, so that's, I'm on that side of the question. Okay, thank you.
Have time for one more?
This may be the last one? Yeah.
I think you'll like this one. Thank you, Dr. Dieffenbach, for being here. I appreciated that you mentioned U equals U in the beginning when you were tracing some of the successes. And particularly in light of the assays you were talking about, I wonder if you could reflect on how U equals U changes the landscape for cure research, because we've had quite a bit of discussion about that this morning. Many people living with HIV are very concerned about cure being defined as not being able to pass HIV on to a partner. So could you reflect on that for us?
I'm not sure I exactly understand the question.
I think so much of the research is around looking at either, as you said, the two roads of eradication versus ART-free remission. Right. And yet, when we talk to people with HIV, sometimes their thought is really, I mean, certainly eradication would be great, but in the short term, people are really concerned about not being able to pass HIV on. So in the context of U equals U with therapy, right, why should we pursue...
So then you undergo a structured treatment interruption, you're virally suppressed, and you have this rebound, and then you suddenly have virus in your body again, and you could potentially transmit. That's why I was making the point about
even
the
just
assay.
Without therapy, right, that maybe the threshold is higher than that. You know, you may still be able to pass it on, even though you're theoretically controlling, right?
Well, that's an interesting question. I mean, we have had, so remember the 052 trial had a cutoff of 400, because that was as good as the assay. So if you had a viral load under 400, you were essentially declared undetectable. Now we're talking about under 20 or under 50. So is there a window there that is still okay? If you go back to some of the early work with other regimens, when cutoff was 1,000, there's at least one case where somebody had a viral load over 1,000 where there was a transmission event, and it was in the first 6 months prior to full establishment of control. So there may be actually a greater forgiveness. If you think about, and this gets into a mathematical argument, if you think about 1,000 copies per milliliter and the amount of blood that is usually around, I mean, that's like one virus particle per microliter. I mean, so it's not a whole lot of virus. So I think we just have to think about it from the standpoint of when somebody is not taking therapy, that they're monitored as closely as possible. At the same time, here's another role for behavioral and social science. Is there a way to make sure that partners are engaged and participate, not in a bad way, but in a way that they're equally informed? And so the partner knows that PrEP is an opportunity for them during the period of their partner being under a treatment interruption. But, you know, this is where quality-informed consent and talking to study participants will make a big difference. Thank you. I'm glad you got me good response.